Millions of years ago, the ancestors of modern cephalopods made a high-stakes evolutionary trade. They abandoned their outer shells for speed and agility. Left with soft bodies in a predator-dense ocean, they traded armor for extreme vulnerability. To survive this anatomical deficit, the subclass Coleoidea had to develop a new defense mechanism. They evolved extreme behavioral flexibility, relying on camouflage and active problem-solving to outmaneuver predators. We can observe the alternative outcome of this evolutionary pressure in the nautilus. By retaining its external chambered shell and physical protection, the nautilus never faced the selective pressure to develop a complex brain. This phylogenetic tree shows the evolutionary split between Nautiloidea and Coleoid groups. The table data reveals massive percentage growth in the octopus's vertical and optic brain lobes, compared to baseline negative values of the shelled nautilus. Morphological vulnerability directly forced the coleoids to adapt. Without a shell to hide inside, they evolved a centralized brain capable of vertebrate-level learning to navigate a hostile environment. In adult octopuses, this neurological hardware is obvious. They are documented performing associative learning, complex tool use, and intricate maze navigation. But there is a significant gap in the ethological literature. We know adults possess these abilities, but it remains unclear when they activate during juvenile life stages. To pinpoint this cognitive emergence, researchers selected the highly camouflaged California two-spotted octopus. This model organism is often used in labs to test the spatial learning capacity of juveniles at roughly 4 months of age. In the wild, these juveniles inhabit rocky reefs and intertidal zones. Because they are heavily preyed upon, their baseline behavioral state during the day is highly cryptic. They stay completely concealed inside tight burrows to survive. This creates a central tension for ethologists. Researchers must design an active behavioral assay for an organism whose evolutionary hardwiring prioritizes absolute concealment. Testing pure spatial memory requires strict environmental controls. Cephalopods have highly sensitive chemotractile systems. If a juvenile octopus finds a target by following a scent trail, it invalidates the test. True spatial learning requires the animal to navigate using visual landmarks alone. This diagram maps the complex holding aquaria required to achieve isolation. Each juvenile is housed separately, while PVC lines ensure identical water chemistry. To eliminate cross-contamination, water cycles through a central sump system, stripping it of olfactory cues. With the environment sterilized of scent, researchers implemented a starvation protocol. The octopuses were deprived of food for 24 hours prior to the trials. This artificial stressor is designed to override their fighting instinct and trigger active foraging. Under these strict parameters, researchers can be confident in the data. Any improvement in navigation observed here must be the result of cognitive spatial mapping, not scent tracking. The experiment itself relies on a spatial maze. The starved subjects are introduced to novel environments that contain specific physical landmarks and a hidden food source. This image displays the medium complexity structures used in the trials. These white PLA rock formations were 3D printed to simulate the natural, escalating barriers of a rocky reef. This hand-drawn diagram illustrates the expected baseline behavior for a subject's first trial. Without prior knowledge of the layout, the octopus relies on chaotic exploration, taking a highly indirect and wandering route to locate the food source. But if the juvenile possesses spatial learning, the route will evolve. This diagram shows the benchmark for success. Over successive trials, the subject recognizes the structural landmarks and navigates a highly efficient, direct path to the reward. This graph models the projected behavioral data. As the octopuses learn the maze over a 16-trial training period, the total navigation time should experience a steep, measurable decline, dropping from an initial 3,000 seconds down to roughly 300. Generating data that tracks along this theoretical curve would definitively prove that early-stage juvenile cephalopods possess functional spatial memory and advanced cognition. In experimental ethology, perfectly controlled models routinely collapse upon contact with wild animal instincts. The first major failure of the study was structural. Before the training regimen could even be completed, unexplained premature mortality wiped out half of the isolated subjects. The secondary failure was behavioral. The surviving octopuses categorically refused to engage with the spatial maze. They ignored the food source and showed almost zero interest in exploring the 3D printed rock habitats. Video evidence recorded during the trials revealed extreme cryptic behavior. Rather than foraging, the juveniles chose to squeeze into crevices and remain perfectly still and hidden for up to 23 hours a day. This graph charts the actual data captured by the aborted experiment. Instead of a smooth, descending learning curve, the completion times produced a volatile, fluctuating line of random results, showing no behavioral convergence at all. The behavioral metrics failed completely. And this leaves researchers with a paradox. It is impossible to tell from this data if the juvenile octopuses lack intelligence, or if they simply refuse to demonstrate it. To resolve this paradox, researchers must bypass behavioral assays entirely. By employing histological sectioning, they can look directly at the physical maturity of the brain tissue itself. Scientists sectioned and analyzed the brains of octopus bimaculoides hatchlings, ranging from 3 to 13 weeks of age. The tissue analysis mapped out distinct areas of rapid tissue development, pointing directly to the pedal, optic, and vertical lobes as the primary centers of growth. The sectioning confirmed that by just 11 weeks of age, the total brain architecture required for higher learning and memory is fully developed and operational. Advanced cognition is undeniably present in these juveniles. The experimental breakdown occurred in the testing methodology, not the animal's biology. This forces a final question. If the hardware for intelligence is fully online in a juvenile octopus, why does the behavioral software crash during spatial testing? The answer lies in the hierarchy of survival. A juvenile octopus's primary evolutionary imperative is to avoid predation. At this life stage, absolute immobility and concealment override every other function, even the drive to forage for food. Standard spatial mazes are inherently flawed for early-stage cephalopods. They demand vulnerable, active exploration in an open environment from an animal whose entire survival strategy depends on staying hidden. Future cognitive testing must pivot to passive associative learning. An assay like this 4-zone food preference test allows researchers to measure choice and preference within a tight, controlled radius, eliminating the need for long-distance navigation. By testing pattern recognition or delayed gratification while the octopus remains in a sheltered state, ethologists can extract accurate cognitive data without triggering the animal's defensive freeze response. True comparative cognition relies on evaluating brain capacity through the specific lens of an animal's developmental